Now the movie should play, actually. Does it play?
Yes, yes, it plays very well.
Okay, perfect, great. So thank you very much for having me here. I mean, obviously, you know, I maybe am a little bit misplaced because I will talk very little about vertebrates, but hopefully I can provide some, let's say, evolutionary perspective, which will sort of show that some common principles which can be applied to any, let's say, animals. So I would like to kind of try to convince you that there is that it makes sense to use a comparative approach to study morphogenesis. And so so we all know that multicellular life has amazing ability to create shapes. We are showing this here for 3 different realms of life, for animals, for fungi, and for plants. And so the question is, you know, how does life do that? How does it create shapes? It's simple. Units of life are cells, and in order to create shape, they have to act in concert. So their action has to be coordinated. In particular, animals are extremely good at creating shapes, and I like to really demonstrate that on a crustacean, which is an invertebrate, which has the special property that on each segment of its body, it has a specialized appendage. This appendage forms a certain function or fulfills a certain function, and it has a specific shape. Now, this shape arises from the process of development. And on the next slide on the movie, we will see development recorded by light sheet microscopy, and we will be able to essentially follow the almost all the cell in this animal 10,000 times sped up, and we will look at the development in a couple of seconds, which lasts over 4.5 days. So as we play the movie, we at some point start seeing these little primordia, which will give rise to the appendages. And as I zoom into this movie, which maybe doesn't play so well, we really see the spectacle of morphogenesis unfold in front of our eyes. The cells are dividing in a certain direction. They are moving with respect to each other, and they are creating this shape in front of us. So how did life and how did animals acquire this ability to really create shape by coordinating a behavior of cells? Obviously, the answer to that must be through the process of evolution. And in fact, we know a lot about evolution. We know that all the animal lives which we saw today originated its multicellularity through one way. It was one event which basically taught cells how to work together. And when we look at the morphogenetic mechanisms which we study today, we understand them very, very well. We understand how animals are related on the levels of genes and genome. We have a perfect phylogeny of animals, and I will talk about that more later on. However, what we do not understand is how did these morphogenetic processes actually evolve. In other words, we know next to nothing about the phylogeny of morphogenesis. And so if we accept the premise that morphogenesis is nothing else than coordination of cell behavior to create shapes, I think it's really clear that we don't know, and we need to know how many ways there are for cells to change their behaviors. We don't know, and we need to know how many ways there are for cells to coordinate their behaviors. And we need to start to understand when and how many times have these mechanisms evolved. So in summary, I would like to argue that in order to understand the multicellular life, its current, its origin and its current diversity, we need to shift our focus not only from the genes and genome, but we also need to understand cells and processes and understand how cells shape, change, and behaviors collectively under the control of the genes. So in order to do that, we need to find a good system how to study that. And in principle, we can use any system where comparative approach is applicable. One of the such systems is gastrulation, and there are a few reasons for that. It is a process which is very ancient, and therefore it is common to all animals we will look at. It is simple and accessible to experimental manipulation, and there is increasing evidence that to some degree it is also a self-organized process which can occur in the absence of stimuli from the outside. So I will, during this talk, give you 2 little vignettes of how we use the comparative approach in the context of insect gastrulation to learn something about how morphogenetic mechanisms evolve, and in fact to discover new morphogenetic mechanisms. It will be focused on gastrulation in tribolium, and we will be looking at generation of forces and at the mechanism of epiboly, which I will explain a bit more. I will start with a story which is published actually a while ago, which, however, really highlights the power of looking at something a little bit unusual in the morphogenesis research. So in morphogenesis, it is always assumed that the forces are generated from within the tissue, that the cells are behaving as active material. They have some intrinsic ability to generate forces, for example, to contract. And one of the mechanisms how that can lead to formation of shape is that they can contract asymmetrically, creating compression on one side of the epithelial sheet, and this leads to invagination. However, what many of these studies neglect is that the cells are not in isolation, they can interact with their environment. It is neglected because cells are in general in, let's say, in invertebrate, quite shielded from their environment by the eggshell in which the gastrulation process happens. However, it opens one possibility for interaction still, and that is the interaction between the cells and between the inner surface of the shell. And so we discovered that this actually plays a very important role. To cut the long story short, gastrulation of tribolium is characterized by this massive unidirectional flow of the cells around the invaginating embryos. And physical modeling, which is based on the on the concept of intrinsic force generation, has predicted that it is impossible to achieve such unidirectional flow without having the system anchored, attached to some external reference frame. And we were able to collect a lot of experimental evidence showing that in tribolium, in the place where the anchor is shown in this part of the embryo, the cells are in direct contact with the surrounding envelope, which you can think about it as an eggshell. It has a name which is called vitelline envelope, but that's not important. And this attachment is absolutely necessary to create this unidirectionality of the tissue flow. Interestingly, we were able to indicate, to collect evidence that this interaction is mediated by integrins, which gave us a molecular handle to start looking back into the system where this is typically studied, which is Drosophila. And so in parallel, in Tomancak and in my lab, we have discovered that also in Drosophila, in the heavily studied process of Drosophila gastrulation, integrin-mediated attachment of the cells to the surrounding necessary for the proper invagination of the hindgut. And that has very interesting evolutionary implications, because in tribolium, in the beetle, which is a very fairly distantly related insect, the attachment happens in the place where another invagination forms in the beetle, which is the invagination of the head. They have nothing in common. They are different parts of the animal. They are mediated by different integrins. The only thing which is interesting is that they are happening at the interface between the embryonic and the extra-embryonic tissue, which indicates that perhaps integrin-mediated evolution of integrin-mediated attachment at the boundary between those tissues provides a barrier for the buckling of the tissue. But from the point of view of this talk, I think the most important point I want to make is that the discovery of this, we have to imagine that Drosophila gastrulation is being studied intensively by many labs for 25 years. And people have not been thinking about it. And one starts thinking about it when you look at tribolium, where the attachment is really amplified and you cannot miss it, right? So this leads me to conclusion that really looking at morphogenetic processes in a non-traditional model species has really enormous potential to lead to discoveries of a new, as yet unrecognized realizations of the morphogenetic processes. So I will now switch gears, but I will stay in the system and I will show you a vertebrate actually, so that you have something to really identify with, right? So you all know the fantastic process of EVL cell spreading during the epiboly of zebrafish. This is a process which is also studied by many labs, and in particular the lab of C.P. Heisenberg has made seminal contribution to explaining the mechanism of this spreading during epiboly. They've shown that this is driven by actomyosin cable in the yolk, and it has been shown also that during the spreading, the cell sheet is really massively stressed, and that this stress has to be relieved, and that the relief of the stress is achieved through asymmetric cell division. Actually. So why am I talking about that? insects, they have also a kind of epiboly. So what I was talking about here is a process where the extraembryonic serosa is flowing over and surrounding the invaginating embryonic rudiment of tribodium. It is schematically shown here. The process was initially very nicely described by Tassos Pavlopoulos and Matt Benton. So this is essentially very similar to the epibolic process in fish. However, there is one important difference, and that difference is that in this system, there is no cell division. So these cells which are spreading, they do not divide. We know that, right? And so there must be another mechanism how the stress in this really, massively spreading sheet, it gets somehow relieved. So we went after that, trying to understand what this mechanism actually is. We used for this light sheet microscopy. This is just a little one slide about the benefits of light sheet, where you can image embryos from multiple angles. You can get beautiful in toto recordings. Those are a little bit hard to analyze because they are large data, but they give us here the crucial ability to look at the embryo from all sides, because this process happens all around the embryo. To simplify things further, we can project this three-dimensional data into two-dimensional cartographic projections. And here, crucially, we can look at the dorsal at the top side of the embryo at the same time as the ventral side, as the bottom part of the embryo. You will notice this closing of a window. This is how it's called. It is called a serosa window closure. It is really this spectacular process, which will start now again, where these cells from the dorsal side initially, they really flow all the way over the posterior of the embryo, and they will eventually close around the embryo which goes inside. That's why in this projection it gains color, and this window will close and will completely surround this embryo. When you have it projected like this, you can very easily extract quantitative information about the cells. You can paint it onto these cartographic maps, and you can really make analysis about the spatial arrangement of the properties of the cells in the entire process, and you can look at it in one go. If you want to be convinced that it actually makes sense in 3D, you can always back project this data back to 3D. But this is one technical thing. Rest I talk about is science. Okay, so what we found out, which is really crucial here, is that as the window which I described, hopefully you managed to gain an idea that the window is happening. The window initially has at its rim many cells, 75 cells, right? But as it closes at the bottom of the embryo, at the end, only 5 cells will contribute to the closure, right? This is quantitatively measured. And so we imaged this with a high resolution, and we discovered the quite peculiar behavior of the cells at the rim or at the edge of the window. The cells, they are shrinking their edge, which is facing the window, and they are gradually de-intercalating in the plane of the epithelium, inside the epithelium. So this is a behavior which is kind of a little bit modified version of the T1 transition, nothing really unusual in epithelia. However, when one looks at it globally, what you notice immediately that this seemingly homogeneous extraembryonic tissue is actually divided into two very differently behaving zones. The ventral zone, the cells are de-intercalating, they are exchanging neighbors, and therefore they behave like a fluid because their units, the cells are dynamic with respect to each other. Whereas the dorsal side of the tissue played again, there is no exchange. So this tissue, from the definition of the fluidity on the cellular level, appears to be more solid, more rigid. So this is, of course, just the indication. We wanted to get something more quantitative. We used a model which has been developed for exactly this type of epithelia to be able to look from static pictures using an index called shape factor to estimate the fluidity or rigidity of the tissue. We extracted the shape factor for the cells during the process, and when we compare the shape factor for the cells on the dorsal and the ventral side, we see that the cells on the ventral side, they show a significantly higher value of the shape factor, which indicates a soft tissue. To corroborate that further, we performed physical experiments where we measured the tension in the spreading epithelium. I'm using the fish schematic here to give you an intuition. One would expect, well, at least I would expect intuitively, that the tension is going to be highest at the place where the cells are maximally stretched. We can test the tension by one way, which is to perform a large-scale laser ablations and measure the recoil velocity after the cut, and like that estimate what is the tension in the tissue. And surprisingly, we find that on the ventral side, the tissue is recoiling slower, meaning that in fact the tension landscape is the opposite, which we take as a indirect corroboration that the tissue on the ventral side appears to be more soft, more fluid-like, whereas the tissue on the dorsal side is more solid. Now, what could induce this transition of the really genetically identical tissue? So we noticed that at the rim of the cable, there forms a very continuous, very prominent cable of actomyosin, which contains both accumulation of actin and myosin. We found that this shape of that cable mirrors the window. It changes from irregular to rectangular to circular until it closes entirely, meaning that the cable also shrinks in size down to 0. We quantified the amount of myosin in the cable, and we found that it doesn't increase. It at best stays constant over time. We measured the tension in the cable by using these laser cutting experiments. In fact, we performed these cuts also multiple times to show that the individual cells at the cable are actually independently contractile units, that the cable doesn't have some global tension. You have to cut it, you can cut it several times and the recoil is always the same. But what was the most interesting and telling about the properties of that cable was that it is highly heterogeneous. So the amount of myosin at the different cells is very, very different. We can visualize that beautifully in these movies, which are projected here and annotated here. And so what looks here is that the cells which have the highest amount of myosin, they shrink the fastest, meaning that they will be more likely to de-intercalate from the cable first, which creates this heterogeneity and the ability of cells to change their neighbors. So this leads us to the model that the cable heterogeneity is inducing the local tissue fluidization on the ventral side in the vicinity of the cable, which facilitates the closure of the window. We would like to test that, but in this system, now this is a disadvantage of working on a non-model system, that you don't have all the tools. It's a little bit difficult, but we can do one thing. We can genetically homogenize the embryo. We can basically make the boundary between the embryonic and extraembryonic tissue go away, and the boundary is what makes the cable. So if we do that, if we do this kind of genetic knockdown experiment, we can show that the cable is completely abolished. There is no cable. Now, if we image this dynamically, what happens is that the window closure begins, but then it stalls and it cannot continue. So we use that as to us only available evidence to indicate that the cable really is required for the local tissue fluidization. It's not super conclusive, but we can discuss about this. Anyway, but from the evolutionary point of view, what is really striking is that this mechanism, which is used in the physiological process of serosa window closure in tribolium, is very reminiscent of a homeostatic mechanism which happens in Drosophila when you induce injury to the tissue. When you make injury in the epithelial tissue in Drosophila, the injury will close, and it will close via a mechanism which involves a heterogeneous actomyosin cable, where cells are actually, this is shown on the right movie here, which I maybe play again, where cells are actually leaving the periphery of the cable. You can see the cable here, you can see its heterogeneity, and the lab of Jan Malmau and others have shown that this induces local tissue fluidization. So this leads me to conclude that local tissue fluidization could actually be a conserved morphogenetic mechanism that is used in both normal development and in homeostasis to close the epithelial gaps. So that was basically the end of science, and I will now, kind of try to wrap the story back to this ground, let's say, evolutionary argument, which is a little bit conceptual, but humor me, this will take another maybe 5 minutes. So in developmental biology, we often think in the terms of a morphogenetic module, which we understand as this kind of very dynamic interplay between regulators, which somehow specify fate of cells. They cannot really make cells do things. This is done by cytoskeletal effectors such as actomyosin, which we talked about. But if the regulators coordinate the effect of the cell intrinsic effectors, you get collective cell behaviors. These backward arrows are probably more important than the forward ones because there are feedbacks in this across all levels. This is example of a morphogenetic module, which is about enfolding of the tissue. And on the right side, even though you might think that's it, this is actually just one realization of it. That is one realization which happens in Drosophila, where transcription factors specify mesoderm, and then they induce somehow asymmetric contractility of the cortex on the apical side, which leads to invagination of the tissue. And so this is a mechanism which is called apical constriction. So people in developmental biology do this for a living for decades. And product of this research is that we have a very deep understanding of many such morphogenetic modules and manage such realizations on the molecular, on the cell biological, even on the biophysical level. But this understanding comes really from looking only at those mostly, not only mostly, at those 4 species which are shown here. What we are not doing, I would like to argue, is using the power of the animal phylogeny. We sample it extremely sparsely, but animal phylogeny is in biology the closest thing we have to a ground truth. It is something we can really lean on, and many areas of biology really rely on that as a strong theoretical framework to make predictions. So I think now it's really a time to exploit the power of the theoretical, solid theoretical framework of animal phylogeny to understand evolution of morphogenesis. In order to do that, we have to expand our horizons and we have to look at animals which are typically not used in morphogenetic research, such as the ones which are shown here. And if I have enough time, I can show you more pretty pictures of those. Now the question is, what will we actually learn when we compare distantly related species? And I really like this example, which is about comparing animals that in sister species choanoflagellates. It's been recently shown by Thibaut Brunet that choanoflagellates, the colonial ones, they also have actomyosin contractivity, but unlike in animals, everything is different. In animals, this contractivity is controlled by transcription factor, whereas in choanoflagellates, it's controlled by light. The purpose of the contractivity in animals is to pattern the embryo and to invaginate mesoderm to do gastrulation. And in choanoflagellates, it is about changing the behavior of the colony from feeding to swimming. The molecular details of how this actomyosin contractility, how this cable appears, are not entirely clear. Maybe it's entirely different in the 2 species. So what do they have in common? What they have in common is the need to bend the tissue. Right? This is here common, and this is essentially physics. So this leads me to a realization that fundamentally biology is an expression of physics. And I'm not the first person by far who have been thinking like that. This is an old idea coming from Entwicklungs Mechanik. However, at that time, even though these people were really pioneers and they thought about developmental biology in terms of physics very clearly, what they were missing was the ability to really visualize this tremendous, incomprehensible complexity of the biological system and to kind of infer on it even without the human bias with the computers. And they didn't have the phylogeny. They didn't have this strong theoretical framework to do evolutionary inferences. So I think now we have those 2 things, and now it's time for us to basically try to understand how evolution exploits physics to create form. Now, in order to do that, we need to build a bridge between 2 large and established research fields, which is evolution of development and tissue morphogenesis. You might want to say, why building a bridge? Isn't that bridge already built? I will convince you that it is not, because Evo-Devo is a research field which is all about the regulation. It is relying on the really universal technique of looking at patterns of gene expression by RNA in situ hybridization. It relies on sequence alignment to understand the ancestry of the sequences, and it is relying on a very strong theoretical foundation in population genetics. Researchers in this field have established beautifully how information transfer in biological systems evolves, and they are using non-model species by definition because that's what the research is about. However, I would argue that not everyone, but many people in that field are less interested in how the genotypes translates to phenotype, meaning that the mechanism remains a black box. But here, this other research field comes to the rescue, because this is a research field which is really trying to understand how coordination of the effectors inside cells leads to collective cell behavior. It is a very quantitative research field, which is using the latest advances in imaging, in physical manipulation, and which is building on the super solid foundation of a physics theory. This is a field which is all about mechanisms, and it is a field which has, in the biological sciences, in my opinion, made the biggest foray into using physics to understand biological systems. However, nothing is perfect, and this field as well is lacking the interest in many cases for establishing the connection between the mechanisms and the biological regulators. And because the approaches are so technologically demanding, there is a very strong model organism bias. So people stick to this kind of evolution, to this toolkit or organisms where they can do this. So I have a couple of more slides, but I will stop on my last slide, on the next slide. I think now is the time to combine the conceptual, the methodological, and also the theoretical approaches of Evo-Devo and tissue morphogenesis and study evolution of morphogenesis. I think this kind of lessons from tribolium and from many other examples I could have covered, but I was focusing on mine, is that we will really, by taking this broader approach at phylogeny, we will discover many new realization of morphogenetic modules. We will discover new mechanisms we simply do not even know about yet. We should be able, with the toolkits which we have, to connect systematically gene regulation to the biophysical mechanisms. And if we manage to do so across the phylogeny, I hope that we will come closer to understanding how biology utilizes physics to evolve new forms. So this is that. I will actually skip to my acknowledgments because I don't want to really waste your time. Do you see the acknowledgement slide? Sometimes it doesn't happen when you jump out.
Yes. Yes.
Okay, great, perfect, right? So first of all, I want to kind of plug here that this was something which we were trying to sell this to a major biomedical organization to start an institution. That didn't happen, but nevertheless, I have recently become a director of Central European Institute of Technology in Brno, and I skipped a few slides where I talked about how we are going to exploit the combination of live imaging and electron microscopy, which gives some credence to the fact that Brno is the place to do it, because 35% of electron microscopes come from Brno. So anyway, that's the future. But this whole idea of looking at evolution of morphogenesis was defined during the competition for the Janelia Research Campus New Research Direction, and I must acknowledge my two partners in crime, C.P. Heisenberg, Andreas Heino, with whom we formulated these ideas, and the beautiful think tank which we were allowed to assemble at Janelia. And finally, I would like to thank the people who did the work on the two vignettes which I described. Stefan Minster did the work on the attachment, and Akanksha Jain did the fantastic work on the epiboly. This was brought to my lab by Tassos Pavlopoulos, and it was powered by the really generous and fantastic sharing tribolium research community. And you cannot do something like that without a bona fide physicist, and we have one in Dresden. We have probably, we say, the best one, which is Stefan Grill. So, and down here is my lab who does all kinds of things. So thank you very much.
Thank you very much, Pavel, for this very interesting talk. Questions from the audience? Nadine, I'm sure you have many questions.
Nadine, I am all ears. I can't hear you.
Okay, just one question related to this link between gene expression and forces, let's say, gastrulation forces during gastrulation. So you say it probably will imply to stick to classical models.
Yeah, I don't know if that's really what I mean, right? I mean, I think that there is definitely a need to do a better connection between the modeling which happens on the, let's say, patterning level, and the modeling which happens on the physics level of force generation and tissue mechanics, right? But I think I would go even farther, and I would say that the theory, multi-scale theories, they could come from different directions. And so what would be really interesting is to also incorporate into that thinking the theoretical modeling, the evolutionary theory. And honestly, this is very easy to say. You could then ask me, how would you do that, right? And the answer, the simple answer from me would be that I don't know. And I don't think that anybody knows. But it would be really wonderful, if we find a place where we could try to figure this out, right? How can we combine the theory from evolution and from physics? Once again, when I say anything like that, I'm not saying an absolute thing, because I'm sure that in the audience is someone who does that, right? So what I'm saying is, like, we need more of that.
Donald, do you have a question?
Yeah. So I guess like your aim is something that's fairly universal, Pavel. I really like the talk, but a lot of your studies and your examples are very epithelial, or into basically they deal with thelia, and not say something that's like mesenchyme, especially like in amniotes. You have a lot of evolutionary creativity in things like cartilage and bone, which aren't terribly epithelial structures, especially. And so what kind of interactions do you see, or is this, I guess, like it's hard for me to imagine how to take this to those types of contexts which don't have strong sheet-like structures to the base tissue?
I mean, honestly, it's an interesting question, and thank you for that. I mean, I haven't really thought about that much explicitly, but I don't really see conceptually there would be any difference, right? I mean, there are theories. I mean, Auger-Campas is looking at tissue fluidization in mesenchymes, Heisenberg as well, right? So you can totally, I mean, there are theories, percolation, whatever, I don't know, right, which are really completely relevant for non-epithelial tissues, right? So I think that even even that division might be a little bit artificial, because the underlying physical mechanism might really not care so much about how the cells make junctions, right? So I would, maybe that would be my answer. Is to really maybe abstract even beyond that, and say, well, can we found some common principles of how cells organize to create form, regardless of whether they are epithelia or mesenchyme?
Thank you.
So sorry, Pavel, I'm back online. I mean, there was an interruption at the bad moment. My question was about your first point that the cells needed an anchor to properly perform gastrulation movement. So I wondered about this anchor in other species. What would be the parallel or the related structure that would do the job?
Yeah, thank you. Yeah, exactly. So that's a really good question, and I think that's basically what needs to be done next, right? So once again, this was news for insects, right? There are other species where this plays a role. In chicken, it's definitely playing a role. You would imagine that in mouse, you would probably think that this most likely doesn't play a role in terms of zona pellucida, I guess, right? But in many other places, we don't know, right? And so that's where I would say my kind of, let's say, extremist comparative approach would be to really go now across the phylogeny and ask that specific question: in what gastrulation systems, which we can get our hands on across the phylogeny, does such interaction play a role? And we can do relatively simple experiments by removing the shells and seeing, whether morphogenesis happens. And we are starting to do that. I didn't show these slides, but it's a beautiful species of bryozoans where the single cell has hundreds of protrusions with which it hangs on to something which they call a fertilization envelope. And nothing is known about the mechanism of it, but it is needed so that after the morphogenesis happens, the circular envelope can be kind of adjusted to because it will protect the larva. It can be adjusted to the new shape of the animal, right? So this there, it's very obvious that it happens. And so I would map it across the phylogeny, and that's really something which we are actually trying to do.
Okay, well, that would be great. Thank you.